Thank you for this kind introduction. Well, I will be brief about the goals of the workshop. It's clear that the topic, as has been already announced, is on biologicals and biosimilars, which is indeed a hot topic these days, and already actually in Europe for a few years. So the idea of this current workshop of this morning is to give you an insight in the basics of a biological on the one hand, and on biosimilars on the other hand, in order to be able to see the introduction of biosimilars within a context of a safe implementation and a safe use. And you cannot use a medicine in a safe way if you don't have sufficient scientific background on the development of the drug and on how the concept of the production and of the regulatory requirements is situated. So I will start off with some basic definitions on biologicals, give you the key concepts, then have some thoughts about the quality requirements from a regulatory point of view, and then our colleague Mark Till, who is present, will also deal with quite a number of some clinical issues, and then I will also come back with a few other practical stuff. So I will start off. I don't have a watch here with me, so if it's around 10 o'clock, you will have to stop me for a coffee break if that's needed. So the first thing you have to realize when we discuss biological medicines, it's totally different from a chemical medical product. And that is first of all because a biological medicinal product is produced by a living system, and we will depend in part on the behavior of this living system, for which we don't have full control of its behavior. And just to remind you, your biochemical courses, you know that when we have a protein that is being produced, a biological that is being produced, this means that we have somewhere in the cell that does produce this protein, we have the genetic information. This genetic information is transcribed to messenger RNA, and then this messenger RNA is translated into a protein. And it's important to realize that if you recall, obviously, what the properties are of a protein, it's a sequence of amino acids. Different amino acids in a particular sequence will determine the basic properties of a protein, but it's not merely the sequence, it's also the way how this chain of amino acids is folded in a three-dimensional way. This is very important for the functional properties of a protein. And as we will see, it's not only just the amino acid chain and the folding, there are also some other changes that do occur after these amino acids have been assembled and folded correctly. It's also important to realize that we use different types of cells depending on the product, depending on the company, depending on the process. So this is actually the consequence of the fact that the genetic code, the conversion of a DNA sequence to an amino acid sequence, to the protein, this is universal throughout the entire living world. This means bacteria, yeast cells, human cells, they all use the same dictionary to translate the genetic code to an amino acid code. So we can express, in theory, any protein in any kind of cell. Insulin, for instance, a human insulin can be produced in a bacterial cell, for instance. So when we make a biological protein, this also means that we have to generate a cell that will produce, if we want to produce insulin in bacterial cells, there are no bacterial cells in nature that do produce human insulin. So we will have to construct this particular cell line. This is also one of the important processes within the development of biological. And once we have this cell that is very well characterized, then we can start the production of the protein. So from a research perspective, and obviously also from a pharmaceutical company perspective, if we are talking about biological medicinal products, this is the very important crucial starting point. This is your production cell line, which will be kept as a master cell bank, which I will not discuss now because of limited time. This is very important. You have this, this is the, let's say, the very crucial information from which you will have to start, not only today when you want to register your product, but also next year, within 10 years, and hopefully, if you're successful, also within 20, 25 years. So this cell has to be stored in a very stable way, and it also has to be cultivated, multiplied for a production run. We will store those cells in vials of approximately a few hundred microliters, a few millions of cells, and for each production run, we will have to thaw a new vial and multiply the number of cells from a few hundred microliters up to bioreactor tanks of approximately 1,000 liter, 10,000 liter, up to 40, 50,000 liters. So this is an enormous number of multiplications. And that's already a very important point within the process. This is, you have to control the multiplication in a very well-controlled manner in such a way that you do not introduce mutations during this multiplication effect. You will understand that when a cell has to multiply itself, the DNA has also to be multiplied, and with each multiplication step, you theoretically have the chance, not only in theory but also in practice, to introduce a mutation, and that's something you definitely want to avoid. This just to illustrate you the complexity. This is what you see here is actually not a bioreactor tank, because the bioreactor tank is below the floor. This is a small one. I'm just showing you this one to illustrate the complexity of the controlling of the process. All those tubings, they are not just there to make the bioreactor look fancy. These are all tubings and sensors to check the oxygen, to check the glucose, to check the foaming percentage and so on, in order to be able to adjust all these parameters, the pH, the oxygen, the glucose, in such a way that you keep the conditions under which you multiply and culture the cells, that these can be strictly controlled. And obviously, once the cells have produced the protein, you will have to purify the protein from this mixture of cells and other components that have been produced by the cell. And this is just again on an industrial scale. They will even use much larger columns with a diameter sometimes of let's say 1 or 2 meters to get the protein purified. And once you have the protein purified, obviously you will have to characterize it, you will have to check the functional properties, you will have to check the stability, and so on. And as you probably are aware of, the pharmaceutical industry is, I think, within the world, if you look at all kinds of industry, the industry that is subjected to the highest standards of controls. This is not very surprising if you look at the impact that the quality of a medicine has on the society and on the patient in particular. So the different steps have to be shown to be fully under control. This is at the level of the cell bank, as I mentioned. This should also be characterized in very much detail in order also to be able to trace back 20 years later whether you're still dealing with the same genetic composition of this particular cell line, for instance. Then obviously during the entire process, the different steps, up to 100, up to 200 different purification steps, concentration steps, and so on, you have to validate the process in very much detail. And then obviously once you have the purified active pharmaceutical ingredient in bulk, you will have to demonstrate its biochemical properties, its physicochemical properties, stability, and so on. Obviously, you have to formulate your active pharmaceutical ingredient into the drug product to be administered to the patient. This is again an extra manipulation that is required on the one hand, but it also has a consequence that you have to repeat quite a number of characterization steps in order to demonstrate that after the formulation you still have the protein in its form that you expect it to be, and that is expected to have a clinical efficacy. Now, I mentioned that a biological drug will be different from a chemical drug because of the production process, because we depend on this living cell system on the one hand. There are quite a number of other characteristics that differ between a chemical drug and a biological drug that will be important when we discuss the situation of biosimilars. And that is, first of all, the size. And for those who are in the front may see it, but if you look at a chemical molecule, this is, for instance, acetyl salicylic acid, the active pharmaceutical ingredient of an aspirin on the left-hand side. This is, the sizes are drawn to scale. We have then here interferon, a relatively small biological molecule, and then if you look at the monoclonal antibody, the size would look like this relative to this very small size of a chemical molecule. So, and the size difference is approximately 1000-fold between a typical chemical molecule and a complex monoclonal antibody. I mentioned the synthesis in cell culture. This has also as a consequence, a chemical product is very well defined. Acetyl salicylic acid, there's only one molecule type acetyl salicylic acid. Nobody will discuss about different forms of acetyl salicylic acid. This is completely different for a biological molecule because we are dealing intrinsically with a heterogeneity within the population of the molecules. And that's because the structure and the properties do not only depend on the amino acid sequence, but also on some changes that have been introduced during the production process of the molecule. And it's also such that, as we will see, that the heterogeneity within this protein population will be also very much dependent on the culture conditions, the storage conditions, the formulation, and so on. And this is just a textbook example illustrating what kind of biochemical reactions contribute to this heterogeneity. And this is just exemplifying the glycosylation, which means that on particular positions at the amino acid chain, the cell will attach sugar or sugar-like molecules to the protein. This is important. This may have an impact on the stability of the protein. This may have an impact on the solubility of the protein. And as we will see for monoclonal antibodies, this also often has an impact on the functional properties of the protein. So this is an important process that does occur and that is often required to have the expected properties. Now, this glycosylation is a biochemically controlled process that occurs in many different steps. And without going into detail, these multiple steps end up in something like this, for instance. This is not the only possibility, but you will see that each of these arrows represent an enzymatically controlled biochemical process that occurs within the cell. And as you may recall from your biochemical courses, the speed by which an enzyme works is very much dependent on the conditions. This also means that the relative amount of these different intermediates and of the possible end product will also very much depend on the relative speed of these different conversions. And this different speed will be dependent on the culture conditions, for instance. So this illustrates that, at least for glycosylation, for instance, the pH, the amount of metal concentrations that are in the cell, will have an impact on the relative occurrence of these different intermediates and will have an impact on the type and the extent of glycosylation and these different forms. Now, glycosylation is only one of the possible enzymatically controlled processes that do occur and that we all classify as being post-translational modifications in the cell. So you do have a number of other enzymatically controlled modifications, and these all depend on the type of cell that you're using. For instance, a bacterial cell will not glycosylate. A yeast cell will glycosylate in a different way than a typical mammalian cell, for instance. So this depends on the cell type and also, as mentioned already a few times, on the culture conditions. But this is not the only way to introduce variants of your same protein. There are also a number of, let's say, more chemically induced modifications that do occur during the culture conditions within the culture medium, or during the purification process, or during the storage. And a typical one that you may recognize is oxidation. There's always some oxygen in your buffers. It's very difficult to completely remove all the oxygen. Some amino acids in particular, for instance, methionine is pretty susceptible to oxidation. So you will also find some traces of some parts of your protein molecule that occur in an oxidized methionine form. Oxidation, but also a number of other chemical modifications. And these will depend on rather external conditions during the purification, during the storage, and so on. So if you take all these different variations in sources of variation into account, you may end up, in theory, with more than 100 million variants of the same molecule. Obviously, this is the theory. This will probably not occur in practice, but you will easily appreciate that if you are able, from an analytical point of view, to detect at least 20 of those variants, that this will not be surprising, basically. So the point is, when we are discussing biological medicinal products, we know we have to deal with product variants. We have, from a theoretical point of view, a pretty large number of possible variants, and it will be, from an analytical point of view, virtually impossible to unambiguously identify each of the possible variants that are effectively present in your protein population. And so the properties of the molecule are actually determined ultimately not only by looking at the final product, but also by looking at the entire process by which the product has been determined. So the only way to introduce or to guarantee that you have a very limited batch-to-batch variability, for instance, that is to guarantee that the consistency of the production process is really very much under control. And that's why you will often find the paradigm that it's the entire process that will determine the properties of your product. So what does this mean in consequence? What does this mean in real life? Well, actually, if I want to produce a protein in my research lab, I will be able, hopefully, to clone the genetic information of this protein here indicated in green. I will put it into a right genetic environment in a vector, and I will put it into a cell. I will develop my protein, my process to produce it and to purify it, and I will get the protein. If somebody else, my competitor, as a researcher, wants to do the same, or as a pharmaceutical company, I will obviously not tell them everything I know. I will not disclose, I will not give my cell line that I have been working on for quite some time. I will not disclose my production and my purification process. The only thing they can rely on is the DNA sequence that will be publicly available. But they all have to develop their own cell line with their own characteristics. They will have to establish their own production and purification process, and they will definitely also end up with a protein of a very good quality. A quality that is, strictly spoken, comparable to mine and to possibly another third party who decided to produce this same protein. And if you look at it, we all made the protein, as you see here. But if you look, this is at the first glance, if you look into a little bit much more detail, and also if we will look into a little bit more analytical detail, you will see here that the word protein is not exactly the same. So it looks very much similar to each other, but it's not exactly the same. So just as a visual illustration that even though we start from the same, exactly the same genetic information, we will all end up with the protein, but not exactly under the same form, under the same heterogeneity, and so on. In addition, another important point is that for biologicals, when they are administered to patients, they are intrinsically immunogenic. This is much, much, much less the case, as you know, for chemical drugs. So this is also something, when we use biologicals, we have to take this into account. That for some biologicals, it's very well known that they do introduce an immune response within the patient, even if we are talking about a fully human molecule. So, as a real-life visual illustration again of this heterogeneity, infliximab, one of the first, or the first monoclonal antibody that has been registered, that has been approved in Europe as a biosimilar, was infliximab, and this actually meant that, there are two companies who produced a copy of infliximab, and this actually means that even though, if we consider the first line here, the reference product, the originator infliximab, the copies that will be available, or that are available within Europe, have been generated, have a very good quality, have the only thing in common is the cDNA sequence that is publicly available. They all started from exactly the same sequence, but we will see also, if you look at the European Public Assessment Report, and I will come back to that later, that these infliximab preparations are not identical to each other. And let me come back now to the concept from a regulatory point of view of biosimilars. Approximately, well, a bit more than 10 years ago, the European Medicines Agency was confronted with the situation that after the patent had expired for somatropin, there were companies who wanted to make copies of somatropin in a decent way onto the European market. And so they figured out that indeed these copies of somatropin were not identical to the reference product. And then the question is, if they are not identical, are they comparable, or maybe they are not comparable at all for whatever reason. And if we say identical, comparable, then we need to have some criteria at certain levels. And obviously we will have to look at the physicochemical and in vitro biological properties. We will have to look at the impurities on the other hand, and obviously we will also have to have some idea on the clinical properties of the copy that is being generated. And obviously when we want to make a copy, we actually also want to introduce it onto the market, usually with a lower price. So this also means that we will have to see where can we make any kind of shortcut to have the process shortened and in such a way be able to offer it at a cheaper price. And taking all this into account, that's where the European Medicines Agency was the first worldwide to come up with the concept of biosimilarity, referring to the fact that we are dealing with a biological and that we are dealing with something that is similar but actually not identical to the reference product. And I use the reference product because indeed when you need to compare it, you need actually to have a reference product, and you will have to set particular criteria. And if you look, this is just a simple definition from the website of the European Medicines Agency. You will see here that it actually a biosimilar is obviously a biological product, and it contains a version or a copy of an active substance that is already authorized at the European Economic Area, and you will have, through a extensive comparability exercise, you have to demonstrate that your version is actually sufficiently similar to the reference product. And we have in Europe guidelines, as I mentioned, since 2006. So we have quite some experience within the European area with biosimilars. In the meantime, as you probably know, also the WHO has guidelines, and most, or not most, well, many of the other regions within the world also have established their own guidelines with respect to how to develop a biosimilar, how to approve, or how to come to a high quality approved biosimilar. I won't go into detail with all those different guidelines. Overall, there are a few differences that may be important. I will come back to that later. But overall, they are very much the same, and it's all, as you will not be surprised of, it's all about the quality of the product, the safety of the product that you will administer to the patient, and last, but definitely not least, also the efficacy of the medicinal product. And all those guidelines include instructions to compare very extensively the intended biosimilar with the reference product. And obviously, it's very important. You need to have a comparison with the reference product that has already been authorized in a region for which we can expect a good quality product. And this is a pretty busy slide to illustrate to you that at some levels there are differences between important guidelines, and for other aspects the guidelines are exactly the same. And I won't go into too much detail at this moment. But I will draw your attention, first of all, the similarity concept, it's here in different colors, but actually, as I mentioned, it's overall more or less the same. What you will see is, for instance, extrapolation, and we will come to that later this morning. Extrapolation is a concept that is very well recognized in all the different guidelines. And the same for immunogenicity issues that we will have to evaluate for the biosimilar. And also to a certain extent with respect to pharmacovigilance. Differences that may be important is naming issues, and naming is related to traceability and to pharmacovigilance. So it may be a boring issue to discuss about naming, but it may have an important consequence on the floor. And another very important point is the discussion about interchangeability, substitution, switching, and anything that is related to subjecting the patient to either the same molecule throughout the treatment, or whether you would decide to switch from one version of the molecule to another version of the molecule. So there is clearly a huge and important distinction between, for instance, the European Medicines Agency and the FDA, and we will come back to that later this morning. Now, how will you have to start generating or developing a biosimilar? This is also a typical visual illustration. The point is, as a company who wants to develop a biosimilar, you will have to, as I mentioned, develop your cell line. You will have to develop your process. And once you have done this, let's say for the first time, you have produced your cell line, purified the product, you will have to compare your product with the reference product for very different, many characteristics. Since you have the first time to make a guess on how you will have to develop your process, your production, purification strategy, you may be sure that you will end up with a product that is, for certain characteristics, not sufficiently similar to the reference product. You shouldn't panic. You will just have to think, what are the differences? And then in a rational way, adapt your process in such a way that these properties for which there is a difference, which will probably not be acceptable, these differences will become smaller as a consequence of the adaptation of your process. So you will have to go back to the lab, adapt it, do another run, and based on your analytical data, look whether you get closer. After a second run, this may not be sufficient yet, so you may have to do a few back and forward steps. So it's a reiterative process to optimize and adjust your process in such a way that you come closer and closer to the properties from an analytical point of view to the properties of the reference product. And I will skip this one. And once you have demonstrated that from a physicochemical point of view, from an in vitro biological characterization point of view, your biosimilar is very much comparable to the reference product, then if that is the case, then we will assume, or we can expect, that the preclinical properties, that the safety properties, and that the clinical properties of your copy will most likely also be the same relative to the reference product. So everything that happens after the quality evaluation will only be not to prove the efficacy per se, but just to confirm the biosimilarity also at the clinical level. So that's the basic of the concept of a biosimilar. If you have a very solid physicochemical and biological characterization in which it has been demonstrated that your product is highly similar, whatever highly similar means, this is also not fully defined, of course, but when your product is highly similar to the reference product with respect to the in vitro properties, one will expect it to be very similar at the clinical level and at the safety level. So I'm not sure what time it is now. Yeah, so I will continue with the second lecture that is on the program and which will deal with the rather quality aspects and other requirements to get your biosimilar approved. So this is an overview of the guidelines from the European Medicines Agency to get guidelines for the pharmaceutical industry to which they have to adhere or take into account when developing a biosimilar. We have an overall guideline. Then it's important to realize that there is a general guideline on the quality evaluation of your biosimilar. There's also some general guidelines on the non-clinical and clinical evaluation of your biosimilar relative to the reference product. And what is also important, and a little bit of a difference between Europe and the US FDA, for instance, is the fact that in Europe there are also some product-specific guidelines with respect to the non-clinical and clinical evaluation of the different molecules. And that's because you may understand that when we look at a simple molecule like erythropoietin, we may have to look at it in a different way compared to, for instance, a complex molecule such as a monoclonal antibody, because the relationship between the structure and the function for erythropoietin is much more simple than for a monoclonal antibody. And these guidelines will also take that into account. And if we look at a monoclonal antibody, for instance, the guideline will ask or will tell the companies that they have to look, first of all, that the binding of your antibody, the biosimilar antibody, the binding to the target has to be compared with the binding of the reference product to the target. But as you may know, the functional properties of an antibody do not only depend, in many cases, do not only depend on the interaction between the variable domains that are indicated here on top of this structure and the target, but will also depend on the constant region and the physicochemical properties of the constant region. And this constant region, the Fc part, is responsible for binding to quite a number of different sets of Fc receptors that do occur on the cell. So you will also have to demonstrate that your biosimilar infliximab, for instance, does not only bind to the target, TNF, but you will also have to demonstrate that your infliximab biosimilar binds in a very similar way to the different Fc receptors, in particular in vitro cellular models. But obviously, you want your product not only to bind to the target, you will also have to demonstrate that subsequent to the binding of your biosimilar to the target, this biosimilar induces the same effect that you expect. For instance, in the situation of infliximab, it binds to TNF-alpha, but it also has to neutralize the activity of TNF-alpha. So you will also have to demonstrate that your biosimilar copy, apart from binding to the target, also, for instance, neutralizes the target in the same way and to the same extent as the reference product does. And obviously, the same for the Fc-mediated functional effects. You will also have to demonstrate that your copy, your biosimilar, induces the same kind of ADCC, CDC, and so on upon binding to these Fc receptors. So it's not just looking at binding, it's looking at binding to the different regions and actually the different functional consequences upon this binding. So this makes life for a monoclonal antibody biosimilar a little bit more complicated than, for instance, for erythropoietin. That's also something one has to take into account. Now, if we look very briefly to the registration requirements of a medicine, this is for an original product. You obviously have a full quality, a full non-clinical, and a full clinical dossier that has to be submitted. That seems obvious to all of us. For a biosimilar, as indicated, we want to make some shortcuts, but some shortcuts that can be explained, of course. One important point is that when you have an approved biosimilar, you should realize that it has been approved based on a full quality dossier. There are no shortcuts with respect to the quality aspects of your product. And in addition, as you know, you also have to provide an entire set of data that demonstrate that the biosimilar is indeed sufficiently comparable to the reference product. And once that is the case, as I already indicated, the amount of non-clinical data that you will have to provide will be much less because we have extensive experience with the reference product. And if the reference product, for instance, is expected or has no off-target side effects, for instance, we may expect it to be the same for the biosimilar on the condition that we have sufficient comparability data at the quality level. So what stays in dark blue is something that has to be included in your biosimilar registration file. What is in gray is usually not needed anymore. And what it may be important, and we will come back to that later, that if we look at the clinical part, you may notice that it may be sufficient to demonstrate biosimilarity at the clinical level in one indication for which the reference product has been authorized, and for the biosimilar you may get approval for all the different indications for which the reference product has been granted an authorization. This is the concept of extrapolation, which will also be discussed later. It also has an important implication for the use of a biosimilar. The fact that we have, first of all, less clinical data, not only in less indications, but also usually in a smaller number of patients, implies that we will need some more post-marketing information once a product has been approved. So we may feel the need to gather more safety data in larger populations, in other disease indications have more confirmation of the efficacy. And what is also very much important, on the long term, larger populations, we may have to confirm the real comparable immunogenicity, which is usually impossible to identify or to detect within a short term in small study populations. But again, this will be discussed later. So, where are we in Europe at this moment? Just a brief overview. There are a few products that have been refused or have been withdrawn, all for the same reason, because there was something, let's say, wrong with the quality evaluation of the product. Differences that were not acceptable, differences that were too large. In some cases, differences with respect to the efficacy of the product, and so on. Just to illustrate that this process is pretty stringent, and you need to adhere to the guidelines and have a good quality product before it can be approved as a biosimilar. And you may notice that even a relatively simple molecule such as filgrastim, here the pegylated form, there are actually a few also recently that have not been approved yet at this moment. Here's some details why they have been produced. For instance, I mean, insufficient details on the entire process, which just means that we cannot trace back the quality of the entire process, and so on. Insufficient quality, for instance, with respect to comparability. Here we have some differences, important differences with respect to impurity, for instance, a lower efficacy for this proposed biosimilar of interferon alpha-2A, which also demonstrate that they look at all the different aspects. There are approximately 41 dossiers that have been approved as of today, basically within the Europe or by the European Medicines Agency. What may be important to notice is that you may see that for quite a number of products, for instance for filgrastim, we have 4 that were approved in 2008, another 2 in 2009, 1 extra in 2010, for instance, another one in 2014, which illustrates that we will have to deal in real life with different biosimilars from different sources. And this is also something that will have an impact on the floor, as we will come later in this morning session. Another important point is that may make life a little bit more difficult. That is, if we have, for instance, here 4 times filgrastim, and we have here 6 rituximab. This does not mean that there are, for instance, for the rituximab, 6 different sources. You know, and I'm not an expert on commercialization of pharmaceutical products, but sometimes a company introduces 4 times the same dossier to have it marketed under different trade names, but it's actually the same product, just with another label on it. For instance, for the 6 rituximab, these are actually 4 products that are exactly the same. These are all biosimilars, but the same biosimilar from the same manufacturer, basically, but will be marketed under 4 different trade names. And the other 2 are also, again, from another manufacturer, but again, 2 identical process, which again will lead to some complications when implementing biosimilars and when discussing switching or interchangeability issues under certain conditions. Just to illustrate you that we have quite some experience within Europe with biosimilars, but also to illustrate that there will be some complex issues arising when implementing them. It's still not finished yet. We have approximately 13 files that are currently under review. What kind of tests do have to be submitted for your registration? And you see here a few. We will not discuss all of those. It's just to illustrate you some physicochemical analysis. And you see here N-terminal sequencing, C-terminal sequencing, disulfide bonds, circular dichorism to get some idea of the secondary structure. Some size exclusion chromatography to check the number or the extent of aggregation of your protein, which is also an important parameter. Some isoelectric focusing. Glycosylation, as you can see here, and glycosylation is also a complex procedure and will require different analytical approaches to evaluate the type of glycosylation, the extent of glycosylation, the position of the glycosylation, and so on, all to demonstrate that your biosimilar is glycosylated to a very similar way as the glycosylation of the reference product. And yeah, I mentioned this already, in particular for monoclonal antibodies. This is a bit more complex. I will not repeat this now. Yeah, this is just, again, taken from the European Public Assessment Report for the first infliximab monoclonal antibody biosimilar, where they described the specific FAB-related evaluation that was done. As I mentioned here, the TNF binding, SF for instance, TNF not only the binding but also the neutralization has been demonstrated for this particular process. Then actually some functional in vitro cellular models to demonstrate the impact of the binding of the biosimilar to TNF alpha. And also, as I indicated, the FC mediated effects on particular cellular systems. And in addition, the company that submitted the biosimilar dossier for the first infliximab biosimilar also actually did some detailed three-dimensional analysis, X-ray structure, X-ray crystallography, to demonstrate the real three-dimensional structure of the molecule. If we look a bit more in detail for these different European Public Assessment Reports, and you know that if you look at the website of the European Medicines Agency, for each approved medicinal product, you can find a summary of the all the properties that have been taken into account to approve the molecule. It's obviously not the 100,000 pages dossier that has been submitted. I mean, it does not contain any confidential information, but for instance, for the biosimilar, you will find in many European Public Assessment Reports the basic information on which the CHMP, the scientific committee, has evaluated the biosimilar relative to the reference product. And there you can find, for instance, or you will notice that they indeed reported differences at particular properties, differences at the level of glycosylation, for instance, differences at the level of oxidation, as we can see here, differences in impurity profile, and so on. For the biosimilar infliximab, for instance, they also noted differences in the extent of afucosylation. And without going into too much molecular details, the extent of afucosylation has a clear impact, this is very well known, on the binding of the FC part to particular FC receptors, and this may have also an impact on the functional properties of your monoclonal antibody in vitro, but ultimately may also have an impact on the overall in vivo efficacy of your molecule. So if you find a difference with respect to this particular quality attribute, this will be what we call a critical quality attribute, because differences in this particular quality attribute may have an impact on the clinical properties of the molecule, for instance. So this just demonstrates, if you look at the European Public Assessment Report, that what I told you this morning, that indeed it will be impossible to make an exact identical copy of a reference product, but you may be able to make a copy that is highly similar to the reference product, but never fully identical to it. And to give you some examples of differences that have been reported, and some of them may lead to some discussion or to some ambiguous interpretation. And as I mentioned already, this lower degree of afucosylation, a lower binding affinity, and they actually also demonstrated lower affinity, lower activity within a ADCC assay, a cellular in vitro assay, to evaluate the extent of antibody-dependent cellular cytotoxicity. Obviously, it's important that when I highlight some differences, this is not the only property, of course, of this biosimilar. You have to look at it in its the entire collection of all the different properties that have been reported and compared to the properties of the reference product. So ultimately, the experts decided that these differences in vitro, from a physicochemical point of view, and from the in vitro functional properties in view, actually it was not to be extrapolated to actually the expected clinical impact that it could have. So these differences are considered to be non-clinically not relevant within the clinical context. We may skip this one. This one is a very important case which illustrates that the evaluation of a biosimilar is not always a black and white situation. So what do we find in the European Public Assessment Report when they describe the approval process and the properties of a biosimilar SB2 versus the reference product of infliximab? One of the typical properties that is associated with infliximab, it's a chimeric molecule, chimeric monoclonal antibody, and all the patients, not all the patients, I'm sorry, but within a period of 6 months, let's say that on average approximately 40% of the patients will develop antibodies against the drug. This is something typical we know for the reference product. So we're obviously not surprised that also for the biosimilar, a number of patients will develop antibodies. So this obviously has to be evaluated during the clinical development of the biosimilar, and they figured out that, and ADA stands for anti-drug antibodies, so this refers to the immunogenicity of the drug, and the patients treated with the biosimilar, there were 47% that had developed antibodies against the biosimilar infliximab versus only 38% antibody development to the reference infliximab molecule. And obviously when you develop antibodies against the drug, this may have an impact, and that has also been illustrated in some cases, this may have an impact on the efficacy of the product. If you have neutralizing antibodies, then obviously your drug will not work anymore or work much less. And if they looked at all the data that were present, for some of the experts, the CHMP is the assembly of scientific experts that do evaluate the quality of the dossier in all its aspects. And you will notice that actually 14 out of 36 of the scientific experts were not convinced that this difference would not have any impact on the clinical efficacy on the long term for this product. But, you know, in a democracy, it's the majority that decides. So there was, if you calculate this, you obviously have seen this, the majority was indeed in favor of the approval, and the product was indeed then approved. I'm not saying that I conclude that this biosimilar is not a good biosimilar. It just illustrates that even the scientists who had information, much more information that we have, were not entirely agreed on whether it should be considered as a true biosimilar or not. Another point is, when you evaluate the clinical biosimilarity, as I have briefly mentioned, you don't have to have a full clinical package. So it's also very important that you select a particular patient population that is indeed representative for the other patient population. And you will also know that depending on the indication, depending on the patient population, you may have also concomitant adjuvant treatments, for instance, such as methotrexate. Methotrexate will also have an immunosuppressive effect. So these data, 47% immunogenicity versus 38% immunogenicity, was also obtained in a patient population that was treated with methotrexate. That has even an immunosuppressive effect. So this actually means that you have no idea whether this difference would even be higher in the absence of methotrexate, for which some indications were approved. So this also raises some, let's say, uncertainty. And for the sake of time, I will skip this one. Again, some differences that were observed, but not assumed not to be of clinical relevance. But you see here again differences in aphycosylation and so on. It also depends whether particular quality attributes are assumed to have an impact on the mechanism of action, yes or no. So it's all on a case-to-case basis. What we will require for infliximab may not be the case for adalimumab, for instance, and vice versa. Now, another important example, again extracted from the European Public Assessment Report, is the, let's say, the way the scientific committee deals with the data that have been provided. So, for an etanercept biosimilar, as before, they have found that a significantly lower incidence of immunogenicity was observed for the biosimilar relative to the reference product. 1% for the biosimilar versus 13% for the reference product. We can say that's very good because it gives you the impression that the biosimilar, in terms of immunogenicity, is better than the reference product. But obviously, we want to have something that is similar, not something that is better. So that's also very important in the biosimilar concept. You have to demonstrate that your product is similar. Definitely not worse, but also not better, because if you prove that it's better, then you're not sure whether you're really working with a similar product, and then the whole concept may not be able to be applied. So 1% versus 13% is obviously a little bit of a problem in terms of regulatory point of view within the biosimilar concept. You may also know, and I won't go into detail again, measuring anti-drug antibody response is also a technological challenge. It also depends on the patient sample, it depends on the drug concentration, and so on. So within the entire dossier, it was decided that for certain time points, week 4 and week 8, probably the data on the anti-drug antibody response were not fully reliable for whatever reason. Okay, yeah. So what they did is they decided that they should reanalyze the data by removing the data from week 4 and week 8. And then they came up with 0.3% versus 0.7%. Instead of 1% to 0.3, and the 13% decreased to 0.7%. Same data, but just removing week 4 and week 8. Obviously, you will realize as a scientist, if I would like to publish data and I will have to tell the editor that I deleted some data to have a more better outcome, this paper will never get accepted. So again, I'm not saying that there is something wrong with the biosimilar, I'm just saying that, from a scientific point of view, there are some doubts about how these data are being looked at. And ultimately, it's concluded that it's premature to conclude anything about the immunogenicity. So you see that there are some data that are not fully. And I will skip this one. I'm almost at the end. Terminology is important because, and definitely also I think in this region, if we talk about the biosimilar, you should be sure that we are talking about a true biosimilar, which means that it is a very good evaluated version relative to a reference product that has already been authorized, for which the similarity has been demonstrated. And we may only detect only small differences. I won't deal too much with second generation, but you know that, for instance, for erythropoietin, we have not only erythropoietin, but also darbepoietin, the longer acting version. So this is obviously not a biosimilar of erythropoietin. This is a second generation or a next generation version. Filgrastim versus pegfilgrastim, for instance. But apart from that, you may also, and I'm sure you're aware of that, they're also circulating some copies of products, a whole set of erythropoietin drugs that have never been compared to any existing authorized drug. So these are usually referred to non-comparable biologics, which means that they have been produced by somebody in some countries, but they have never been extensively characterized compared to reference products. So these are definitely not falling under the category of biosimilars as we discussed right now. And these are just some examples which I can skip. And I won't go into too much detail, but it's definitely such that these non-comparable biologics have never been demonstrated to be biosimilar, and therefore this is a small slide. If you look here very quickly, this is a biosimilar of Eprex, for which you see an isoelectric focusing. This pattern illustrates the different glycosylation forms very briefly, and you see that these different patterns, the approved biosimilar and the reference product, is very, very similar. You can see this very quickly. If you look at the whole set of other erythropoietin copies that have been introduced somewhere in the world, but definitely not in Europe, not in the US, not in Canada, and so on, you will see that the pattern of glycosylation is very much different between those different copies, also illustrating that definitely they are not to be considered as biosimilar. And, you know, these are a few Dr. Reddy's products, for instance, Reditux. Rituximab is a product that is produced in India, but has never been compared to any approved product within Europe or within the US. You will also see here a 17-patient phase 2 study. This is a very limited number of data, obviously, and also in China, for instance, some copies that are not to be considered as biosimilar. So in conclusion, if we're talking about biologicals, in general, we should realize that these are complex molecules, often composed of different domains that are important for the function. We are not dealing with one particular variant, but with heterogeneous protein population. The entire process will determine the ultimate properties of the molecule. We should not consider them as interchangeable, and we will come to that after the coffee break. And obviously, we need quite some post-marketing surveillance. With respect to the experience that we have so far in Europe with the approved biosimilars, is that first of all, the process is very well challenging and very rigorous, controlled. The pharmaceutical quality is guaranteed, that's per definition. But we notice that there are definitely, as we could expect, differences in quality attributes between the reference and the biosimilar. And the actual challenge is to demonstrate that any of these differences have no clinical impact on the product. And as we have seen with those few brief examples from the European Public Assessment Report, there will always remain a number of residual uncertainties, either purely statistically or purely scientifically. Which means that once we have this approved biosimilar onto the market, we definitely will need some more post-marketing data that will be gathered once the product is being implemented for its real use. And with that, I would like to thank you, finish here, and thank you for your